So the first speaker is going to be Rob Burgess, who comes from the Jackson Lab. He's a professor there, and probably many of you know his seminal work looking at the GARS and ARS mutations. Some pretty high-profile papers there. So take it away, Rob.
All right, great. Thanks very much, and thanks for the opportunity to present some of our work here today. So I'm kicking things off after Dr. Bonneman's. So what I wanted to try to emphasize in this talk is basically the reason you need to understand the basic biology of CMT in order to actually develop therapies. So with that, I'll go ahead and start. Disclosures are not very exciting. So I don't feel like I need to introduce CMT. I think many of you are much more familiar with it than I am. But the point that I'd like to make with this slide is that there are at least 100 different genes that are now associated with CMT. And we don't have an approved treatment for any of those genetic subtypes. So in terms of the basic biology, this potentially means that there are 100 different genes associated and therefore possibly 100 different distinct biological mechanisms associated with CMT. So does that mean that we're actually going to need 100 different therapeutic strategies for all of these different subtypes? So not necessarily, and I hope not, but can we actually, again, use some of the basic cell biology of neurons to see if there are subtype agnostic treatments that might apply to many different forms? And, there are a few different strategies that people are testing to do this. One that we're going to hear more about from Augustine Therapeutics later today is inhibiting HDAC6. So the rationale here is that in CMT, as axons start getting sick in the peripheral nervous system, the acetylation of alpha-tubulin in the axonal cytoskeleton starts to decrease. That impairs axonal transport. As axonal transport gets impaired, the axon gets sicker, and you have this kind of positive feedback system. The rationale for targeting HDAC6 is that it's a histone deacetylase, but also deacetylates alpha-tubulin in the axon. So if you inhibit the deacetylase, you can conceivably improve acetylation of the tubulin in the axon, improve axonal transport, and slow down the whole axon degeneration process in CMT. So there have been positive results in lots of different preclinical studies and several different animal models of CMT that make this approach look attractive. But I'll also say the caveat here is that the effect size is modest and results aren't always totally consistent across studies. So the reason for that may be that the deacetylation of tubulin and this therapeutic strategy to improve that is often several steps removed from the kind of core biology of the mutation. So we're sort of tweaking a downstream effect of this, which again has potential benefit, I think, in several different types of CMT, but isn't necessarily addressing the core disease mechanism of any of them. A somewhat related strategy would be inhibiting SARM1. So SARM1 was discovered about 10 or 12 years ago now by Mark Freeman's lab, as well as Jeff Milbrandt and Aaron D'Antonio at WashU. And SARM1 is a protein that actually promotes axon degeneration. And just last week, I was at an axon degeneration meeting in Washington, D.C., and there were at least 4 companies there that were developing SARM1 inhibitors for a variety of purposes, including possibly treating CMT. This is a strategy that's been put forward by several people, including Ahmet Hoke at Johns Hopkins, as a possible treatment strategy for CMT. And the Milbrandt and D'Antonio labs at WashU have produced promising preclinical data using a rat knockout model of SARM1 across to a CMT2A mitofusin rat model. And they did see improvement in that preclinical study. However, Ahmet Hoke has also published a lack of effect in CMT1A mouse models when SARM1 is deleted. And my lab actually has tested this approach in 6 different types of CMT, and we don't have positive results in any of them. So So I think this is still, again, a promising strategy, but I think we're going to need to figure out which types of CMT are actually going to benefit from inhibition of SARM1. So instead of targeting these downstream effects like SARM or HDAC6, can we actually target the core disease mechanism that's going with these different genetic subtypes of CMT? And in order to do that, somewhat obviously, you first have to actually understand what that core disease mechanism is. So the first step towards that is just to identify the causative gene. And in terms of gene identification for CMT, we're doing pretty well now. For demyelinating neuropathies, patients have a pretty good odds of getting a genetic diagnosis, but for axonal neuropathies, we're still actually at less than 50%. So there's still some work to do there, but nonetheless, we've got over 100 gene targets that have been identified over the last 30 years. So we just heard about gene therapies. I'm going to expand that slightly to say genetic therapies to include things like antisense oligonucleotides and CRISPR-based approaches. There's an entire session on gene therapies later today, so I'm not going to go into detail here. Other than to say that even once you know the mutation and once you know the gene that's involved, you still need to know if you want to turn the gene up or turn the gene down in order to have therapeutic benefit. And that requires a basic understanding of the pathophysiology. So as mentioned for GAN, loss of function mutations are typically recessive, and the gene therapy strategy there is just put the gene back, as we just heard. But there are also lots of forms of CMT that have gain of function mechanisms, including 1A and GARS, which my lab works on. And in that case, you need to turn the gene down. So even with these gene therapy approaches, you need to know the cell type that you want to target. You need to know whether you need to turn the gene up or turn the gene down. How much do you need to turn it up and down? Are there adverse effects in possibly overexpressing or underexpressing these genes? So there's still a lot of basic science that needs to be done. In some forms of CMT, the identification of the gene actually leads you very quickly to a possible therapeutic strategy. And an example of that is SORD, sorbitol dehydrogenase, which was identified just a couple of years ago as a cause of recessive axonal CMT. And as we'll hear from a talk in applied therapeutics later today, is now actually already in clinical trials. So, a really rapid progression from gene discovery to clinical trial. And some of that's because the mechanism of SORD is actually very well understood. So it's involved in a kind of glucose metabolism side pathway where aldose reductase changes glucose to sorbitol, and then sorbitol dehydrogenase, SORD, changes sorbitol to fructose. And these patients have really elevated levels of sorbitol, and that's thought to contribute actually to some of the disease pathology. So, kind of classic biochemistry, if you have a buildup of an intermediate like sorbitol, if you inhibit the upstream enzyme aldose reductase, that should be beneficial. And it looks like that's in fact the case. And there were already aldose reductase inhibitors that actually were approved in Asia, for example, for other indications. And those are being repurposed and tried as therapeutics for SORD neuropathy. Another example where again the gene identification led fairly quickly to a possible therapy is TRPV4 and Chikomori-tooth 2C. So this is an ion channel, and ion channels are great drug targets. So you can typically get agonists that turn them on and antagonists that turn them off. So they're attractive candidates for developing therapeutics. But I listed the three papers here that were all published back to back when this gene was identified as a causal CMT gene. And it's worth noting that two of these papers thought it was a gain of function, and one of the papers thought it was a loss of function, right? And if it's a loss of function and you use a channel agonist, you're actually going to make things worse, agonists would make it better, but antagonists would actually make it worse. So you still need to actually know if you want to turn this up or turn this down. And just identifying the gene isn't always sufficient to tell you that. So I want to move now to some work that my lab has done. And the class of CMTs that my lab is most interested in are dominant axonal neuropathies caused by mutations in tRNA synthetase genes. And the first of these, glycyl tRNA synthetase, was identified by Tony Antonellis almost 20 years ago, well, over 20 years ago now. I would like to think that we're actually finally making some progress on understanding this mechanism and developing some therapies. So I won't go through all of the biology that got us here, but during normal tRNA charging, the tRNA and the amino acid bind the enzyme up there at the top. That's handed off to an elongation factor, EEF1A, that takes the charged tRNA to the ribosome for protein synthesis and translation. And what we think is happening with these mutants is that the tRNA binds the mutant synthetase, and then whether the amino acid gets put on or not, it never gets released to the elongation factor and therefore doesn't get to the ribosome. This results in ribosome stalling, and ribosome stalling activates a stress pathway called the integrated stress response that I'll describe in a moment. So we think the real problem here is actually sequestration of the tRNA substrate on the mutant enzyme. So the integrated stress response we identified based on gene expression patterns. So the two real hallmarks of the integrated stress response are a suppression of cap-dependent translation, so normal protein synthesis, and then the activation of a transcription factor called ATF4 that turns on cellular stress response genes. And Emily Spalding, a graduate student in my lab, found the integrated stress response was activated in CMT 2D mouse models, GARS mouse models, by the elevation of ATF4 target genes in motor and sensory neurons in those mice. And Emily just wanted to ask the basic science question of how is the integrated stress response getting activated? So different stressors like viral infection and double-stranded RNA activate, a kinase PKR. Other stressors activate other kinases. So Emily just wanted to know which one of these kinases up at the top was actually responsible for the activation of the integrated stress response in our mice. And our best guess was GCN2 because it's the one that senses defects in translation, and we were dealing with a tRNA synthetase gene. So again, just the basic science question of, is the integrated stress response getting activated through GCN2? Emily took a GCN2 knockout mouse and bred it to our GARS peripheral neuropathy mice. And she found that the integrated stress response was indeed gone in those mice. So we guessed correctly. It is actually GCN2. But even more excitingly, the phenotype in those mice got much, much milder. And I'm going to say this was actually a real surprise. Emily and I were actually both betting that the mice were going to get worse when we got rid of the integrated stress response. So that's why we do the experiment. But in these graphs, and this is published work from a couple of years ago, the blue lines are GARS mice that have an intact integrated stress response. The black lines are control genotypes, and the red lines are GARS mice that are lacking GCN2 and therefore don't have an integrated stress response. And you can see, oops, that was the wrong thing to do. Maybe I don't have a pointer, but that's okay. So hopefully you can see that, body weight is shown there in the first graph. B is a test of grip strength and endurance that we use where we put mice on an inverted grid, turn it upside down and see how long they can hang on. You can see the black line wild type mice can hang on for a full minute with no problem. The blue line there at the bottom, our mutant mice usually fall off in just a few seconds. But the red line, if you got rid of the integrated stress response, those mutant mice were now able to hang on. But even more impressively, they actually got bigger and stronger over the course of the experiment from 4 weeks of age out to 16 weeks of age. We no longer had reduced nerve conduction velocity. We no longer had axon loss, and we had improved nerve morphology and innervation at neuromuscular junctions. So by all of our outcomes, getting rid of the integrated stress response was really beneficial. And we've reproduced that with, hopefully I can play this movie. Am I going to be able to do that? Yeah, perfect. So we've also reproduced this effect using drugs. So this is a mouse treated with a GCN2 inhibitor. This is the first day of treatment. He's got a GARS mutation. You can see he really can't get his hind legs under him. His tail is essentially limp. So this is a mouse with Charcot-Marie-Tooth type 2D. This is the exact same mouse after 10 days of treatment with a GCN2 inhibitor, and he's now running around and actually looks pretty good. It doesn't look perfect, but looks pretty good. And then this is the same mouse, 2 more days of treatment, and next to an untreated littermate. So the untreated mouse is on the right. You can see that he's got those same problems. He's shaky. He can't get his legs underneath him. His tail has very poor muscle tone. And the treated mouse is running around and looking comparatively good. Has much better muscle tone, much better hind limb function, and eventually runs away, and we have to go catch him. I won't show you all the data, but hopefully that movie convinces you that we have actually really great effects using these GCN2 inhibitors. So we're actually trying to develop this into a real drug and a clinical program with help from partners like Alesta Therapeutics, who's here at the meeting. Let's see. So again, without going into all of the biology and all the gritty details, the way we came up with this proposed sequestration mechanism was in collaboration with Eric Storkebaum's lab in the Netherlands. Eric had done a bunch of biochemistry suggesting that the tRNAs were more tightly associated with the mutant enzyme than the wild type enzyme, and other related experiments. But the one that he showed that I thought was really convincing is he made a transgenic mouse in which he overexpressed tRNA glycine. So 13 years ago now, in collaboration with Will Motley, who's here at the meeting, my lab overexpressed the wild type enzyme, so wild type GARs. And when we crossed mice overexpressing the wild type enzyme to our peripheral neuropathy model, the mouse that I just showed you, it didn't do anything towards correcting it. So putting back the enzyme in this case, a gene therapy strategy like that did not work. It showed no benefit whatsoever. Eric took a different approach, and instead of putting back the enzyme, he put back the substrate. He put back the tRNA. And I won't go through all the details of how he did that or all of this data. It's published, but suffice it to say, when he took a mouse that was overexpressing tRNA glycine, bred it to the GARS mouse model that I just showed you, it basically completely corrected the phenotype. It went away almost entirely. And these are behavioral tests and neurophysiology tests and anatomy. Again, by all outcomes, he was able to suppress the phenotype. So we wanted to do this in a more translational way, and working with Scott Harper, who's here at the meeting, Scott's lab packaged the 4 different anticodon tRNAs for glycine into AAVs individually. Abby Tadneff in my lab then treated neonatal mice with those 4 different AAVs, waited 5 weeks, and then used our standard battery of tests to see if it worked. And it actually worked extremely well. So in these graphs, the right-hand bars are wild-type mice that are either untreated or treated with the 4 different tRNAs. And then the right-hand 5 bars are the GARS mutant mice, again, untreated or treated with the 4 different tRNA anticodons. And you can see ACC really didn't work very well. I can explain that if you ask me later. But we got intermediate effects with 2 others, the CCC and TCC anticodons, and the GCC anticodon actually worked extremely well. That's the one Eric used in his experiments also. And again, by this gene therapy treatment, we were able to almost completely suppress the disease. And this was true of nerve conduction, of motor performance, and you can see it here in the nerve histology. Mice treated with the GCC anticodon, for example, look almost as good as wild type. So we're actually trying to develop that into something that goes beyond just preclinical mouse studies. And with Scott, we're talking to companies like Armatus Bio to see if we can actually develop that program as well. So what I want to emphasize here and just summarize is there are general strategies that might apply across multiple forms of CMT, like SARM1 inhibitors or HDAC6 inhibitors, but those strategies are fairly downstream of the actual causative mutation and the kind of fundamental biology. And while I think they're promising and need to be tested, it's also not clear that they're going to be a magic bullet that's going to have a great effect in all forms of CMT. We're going to talk a lot today about gene therapies and genetic therapies. That's almost the definition of getting at the core disease mechanism, and I think that's an extremely promising approach. And, occasionally the disease gene takes you directly to a therapeutic strategy such as SORD or TRPV4. But often there's 20 years of hard work between discovering the gene and actually figuring out the biological mechanism and figuring out a pathway like the integrated stress response or like a tRNA gene therapy that might actually get you towards a therapeutic strategy. So with that, I'd like to just thank the people who did the work. Abby Tedneff did a lot of this work. Tim Heinz and Samia Pratt are here at the meeting with me today. Tim is working on kind of a related project, and Samia is working on a different gene. And I've tried to mention collaborators as we went. So thanks, and I'd be happy to take any questions.